Dr. Eric Aidoo with the Department of Anatomy and Cell Biology of the School of Medical Sciences, University of Cape Coast. I'm pleased to bring to you the male reproductive system as far as its embryogenesis is concerned. So we begin with a question. Sex, what is chromosomal or genetic or gonadal sex? What point are these achieved? So these are questions we will be able to answer by the end of the lecture. So we want to just look at this picture and ask, what is the sex of this individual? Is it a female or a male? So I'm sure you are thinking through, but the point is that you don't expect to have a male with this type of length of penis, okay? And obviously you don't expect a lady to also have something that looks like a scrotum. So this is more like in between the sexes, all right? So let's see how a proper sex can be determined. So you can't just look at an individual outwardly and say this is a male or a female. You may be wrong. Just by the looks, yes, you may be able to tell that the face looks like a lady or a male. But deep within, you may not be able to tell beyond the physical. So chromosomal sex, also known as genetic sex, is established during fertilization, and it is when the chromosome-bearing spermatozoa, which is either X or Y, meets the counterpart of the female egg, which is usually X. So if an X-bearing spermatozoa meets an X-bearing egg, it's going to be XX. So in embryos with XX sex chromosome complex, the cortex of the indifferent gonad will differentiate into an ovary, and the medulla will regress. How about the other way around? When the spermatozoa is Y and meets the X egg, you are going to have an XY sex chromosome. Okay, and in this case, the medulla of the indifferent gonad is going to differentiate, okay, not degenerate, differentiate further into a testis with testicular lobes, okay, like the seminiferous tubule, and then the cortex will rather regress. So this is the opposite of what happens in the female. So at a point where the spermatozoa bearing X or Y meets the egg, which is always X, once there's fertilization, you have established the chromosomal sex or genetic sex, okay? Now, it's usually the presence or the absence of the Y chromosome that you use to determine the chromosomal sex, okay? Not barely the presence of the X. For instance, you can have XXY. So if you have XXY, is it going to be a male genetic sex or a female genetic sex? Because of the presence of the Y, that will be a male. Okay, so you usually look at the presence or absence of the Y chromosome to really substantiate whether a sex is male. Okay, and if there's absence of Y, then obviously it is a female. So the presence of this X and Y chromosome will have an influence on how the gonad, which is quite naive at that time, or what we call the indifferent gonad, will progress, whether it's going to progress to form an ovary or a testis. Then, once the gonad finds its direction or bearing, as in going towards the male or female, then that will also determine how the genital ducts and the external genitalia will develop. Okay, so we will look at genital ducts and external genitalia. Therefore, the lecture is going to be grouped into three main levels. We are going to look at the formation of the gonad. We are going to look at the formation of the duct system, the male and female duct system, and then we're also going to look at the external genitalia, both male and female. So we are going to cover that of the male, and then later on, you will continue with that of the female. So we've talked about chromosomal or genetic sex, that it is established at fertilization as whether the X-bearing spermatozoa or the Y-bearing spermatozoa meets the egg. The Y chromosome contains a substance known as the testis determining gene. Okay, it's actually a protein. Testis determining gene called the SRY gene. The S stands for sex, R stands for the region, and then Y stands for Y, okay, gene, to determine whether the development is going to go along the male line or the female line. That is why I told you that it is the presence or absence of the Y chromosome that will emphasize whether sex is genetically male or female. So it is a protein that influences gonadal cords to differentiate into seminiferous tubules. You will come to realize that these gonadal cords are also known as the primitive sex cords, which are usually naive and then can represent both the male or female at a point until it continues to differentiate. In the absence of the Y chromosome, an ovary will be established. All right. So that is the testicles. The Y chromosome contains the testis determining gene, okay, called the SRY gene. And then that will influence the development of the testis. So what are the gonads? They are the testis or ovaries, and they are derived from three main sources. You have a mesodermal epithelium or mesothelium lining the posterior abdominal wall. And you have the mesenchyme, which is the embryonic connective tissue, and then primordial germ cells. So this mesothelium or mesodermal epithelium will actually be part of the intermediate mesoderm of the intraembryonic mesoderm. The other parts of the intraembryonic mesoderm be the paraxial mesoderm medially, and then the lateral plate mesoderm laterally. So primordial germ cells, which originate in the epiblast, okay? And remember your week 2 has the epiblast and hypoblast. So before gastrulation, you have primordial germ cells migrating from the epiblast, and then they migrate through the yolk sac by the 3rd week, where gastrulation would have set in. Okay, so gonads appear initially as a pair of longitudinal ridges known as the genital or gonadal ridges in the 5th week on the medial side of the mesonephros. So this is the yolk sac. Those are primordial germ cells which are migrating. It means that they came from the epiblast and then they find their way in the yolk sac and then walk along the hindgut all the way towards the gonadal cord at this point. So this is the hindgut, and that is supposed to be the allantoid, okay? And therefore, if we extend this, that will be the yolk sac. So it will migrate, then go through the hindgut, okay, and then move all the way towards this dorsal or posterior portion, okay, to where the gonadal ridge is. So that is the gonadal ridge or genital ridge, which is actually medial to the mesonephros. So the mesonephros is blue, and this one is medial to it. So these dotted reddish spherical bodies are the migrating primordial germ cells, and they are destined to come to this gonadal or genital ridge. Once they get there, they try to incorporate themselves into the primitive sex cords, which are actually coming from the epithelium of the gonadal ridge, okay, and extending towards the medulla, where the mesenchyme is. So again, this is what we find. So that's the gonadal ridge or genital ridge, and that's the epithelium of the gonadal ridge. And this epithelium proliferates and then forms cords, what we call primitive sex cords. So you can see that there are some undulating nature of the epithelium. Okay, it goes up and down. All right. So those are the gonadal sex cord or primitive sex cords, which are in the cortex at this point. So when you come to this one, you realize that they have moved extensively. The gonadal sex cord, which were just at the epithelial cortex, have now moved towards the mesenchyme in the medulla. Okay, and you have the primordial germ cells. This time around, they are blue in color, spherical, and they are incorporating themselves into the cords, the primitive sex cords. Okay, and then at the background is the mesenchyme. So shortly before and during the arrival of the primordial germ cells, the epithelium of the genital ridge formed the primitive sex cord, as I have explained. So that's the epithelium of the gonadal ridge. Those extensions are the primitive sex cords, and they start all the way from the cortex and then move towards the middle portion, which is the medullary region. Okay, so therefore we can say that the primitive sex cords are connected to the epithelium through the cortex and then to the medulla. The gonad at this point is known as the indifferent gonad. And I put here, why is it called the indifferent gonad? And this is because the primitive sex cords are still connected to the epithelium through the cortex to the medulla. Okay, there's no break in them. They are just uniform and connected. As you see, when it has to form the male, you will see that it will cut its connection with the cortex and rather cause the portion which is in the medulla to differentiate, and then those in the cortex will regress or degenerate, and then the vice versa for the female. In the female, the cortical ones will be the ones to differentiate further, and the medullary one will regress. You know, the female one is a bit complex because at a point, all the primitive sex cords will degenerate, and then a secondary sex cord will start from the cortex. When we start with the female, I will expatiate on that. So let's use this very beautiful diagram to explain what we've said so far. So that's the yolk sac. These are the primordial germ cells. And then they move through the allantoid hindgut, and then they get to the gonadal ridge. At the gonadal ridge, you have the epithelium of the gonadal ridge forming the primitive sex cord. So you can see that here, they are extending all the way to the medulla. So that's the medulla, and that is the cortex. Okay, so migrating primordial germs and making their way there. That is the paramesonephric duct or Mullerian duct. We'll talk about it more in the female. That is the mesonephric or Wolffian duct, which is more significant in males, but we'll see what it will form later on. All right. So that's the primary sex cords, okay, coming from the epithelium of the gonadal ridge, and they extend from the epithelium through the cortex towards the medulla. Then the primitive sex cords will hollow out and form seminiferous tubules, which means that they are going to canalize and form seminiferous tubules. And this is for the male. And have this thick tunica abugenia, which is separating the sex cords, which have now become seminiferous tubules, away from the epithelium, which they actually emerged from. Remember, I said that the epithelium of the gonadal ridge is going to extend into the medulla as primitive sex cord. Okay, so you have the tunica abugenia now separating that epithelium, okay, and detaching it from the sex cords that they form. Now, in the female, you have what we call secondary sex cords in the cortex, as I have said, and then the medullary one, the primitive sex cords, will degenerate to form the rete ovary. We'll come to that later. So Sertoli and Leydig cells are very important structures when you are dealing with the male reproductive system. The Sertoli cells are also known as tentacular cells, and they are derived from that surface epithelium of the gonadal ridge, which gave rise to the primitive sex cords, right? That is why Sertoli cells are found within the testis, and they help form Sertoli tight junctions, and to help, you know, the immune system from getting close to the testicular development. All right, so the blood and everything of the fetus is restricted from going through to expose themselves to the naive and innocent testis. You don't want any autoimmune, reaction. The Leydig cells are derived from the mesenchyme of the gonadal ridge. Remember also that the mesenchyme was at the background, deep to the epithelium where the medulla was, there was a mesenchyme. Okay, so that is what will form the Leydig cells. What are the functions of the Sertoli and Leydig cells? Leydig cells secrete testosterone and androstenedione, which promotes masculine differentiation of the mesonephric duct, also known as the Wolffian duct. So sometimes I say that the males are wolves, Wolffian duct, and it also promotes the formation of the external genitalia. Why do I say wolf for mesonephric duct? Because the female's own is also paramesonephric duct, and its other eponym is the Mullerian duct. So there are times you might confuse Mullerian duct with that of what Wolffian duct. As for the mesonephric and paramesonephric duct, yes, they are quite easy to determine, but the eponyms Mullerian and then Wolffian duct, sometimes you don't know which one will correspond to mesonephric duct and which one will correspond to paramesonephric duct. So for me, what I do is to use the Wolffian duct, which goes with mesonephric duct for the male, and I call it the males are wolves. You know, males are actually wolves. And they are always chasing after things. So this the Mullerian inhibiting substance is produced by the Sertoli cells. Okay, as the name implies, Mullerian is for the female. So you are trying to inhibit the Mullerian duct from forming by producing the Mullerian inhibiting substance, MIS. And this suppresses development of the paramesonephric duct, also known as the Mullerian duct. So now you see why Wolffian male is a wolf can help you to remember that Mullerian is always linked to the female paramesonephric duct. So the Mullerian duct or paramesonephric duct, as we see in the female lecture, female reproductive system lecture, is going to form the uterus and uterine tubes. Then, if everything goes on well, the gonad, the testis is formed, and then the seminiferous tubules are also formed from the primitive sex cord by canalization. Okay, and then also the Wolffian duct helps in the formation of the efferent ducts and then epididymis. You are going to have this formation ongoing. So here you have the type A duct spermatogonia, type A pale spermatogonia. So the duct spermatogonia that gives rise to the pale spermatogonia, which will in turn give rise to the type A pale, more of them. And the type A pales are the ones that are going to continue the journey to form the spermatozoa by forming spermatogonia first. Then they go through the process of primary, spermatocyte formation, secondary spermatocytes, early spermatid, late spermatid. So we want to look at the male genital duct. I told you that we are breaking the lecture into formation of gonad, duct formation, and then external genitalia. So at puberty, the solid gonadal cords acquire a lumen and become seminiferous tubule as they canalize. Once the seminiferous tubules are canalized, they join the rete testis tubules, which in turn enter the efferent duct. The mesonephric duct, which is the Wolffian duct, form the efferent duct, epididymis, and then ejaculatory duct. It is the male duct system. So this is the testis. These are the seminiferous tubules that are being formed from the primitive sex cord or the gonadal cords. Then you have these straight, tiny region known as the straight tubules, which will now connect the rete testis, and the rete testis will connect the efferent ducts, which are formed from the mesonephric duct. Then you have epididymis, which is also formed from the mesonephric duct. Then you go all the way to the ductus deferens, and then on you go to void the content into the urethra, and then it comes out. But obviously, it's going to pass through the prostatic region, prostatic urethra, before it gets to the penile urethra. So I made mention of the importance of the tunica albuginea as a landmark or a significant feature that separates the testicular tubules from the epithelium. Okay, so or the covering epithelium there. The tunica albuginea is the characteristic feature of testicular development. It separates the tubules from the covering epithelia. Then we will look at the male external genitalia, which begins in the form of the phallus. It enlarges and elongates to become the penis. The urethral folds form the lateral walls of the urethral groove, which is usually found at the ventral surface of the penis. Okay, then the urethral folds will fuse with each other along the ventral surface of the penis to form the spongy urethra. This is what we are talking about. So at this point, that is the phallus, which will form the penis. The middle portion is the urogenital membrane. Then you have the urogenital folds. So ideally, if everything goes on well, the phallus is going to form the glans penis. And then you have the penis also there connected to the glans. Then you have scrotum, which is actually going to come from the membrane here, the labioscrotal swelling will form the scrotum. Then eventually you have this midline showing you where the merging occurred. This is for the female. I don't want to go into that. We'll deal with that when we look at the female. So again, that's the phallus. That is the urethral fold. That's the labioscrotal swelling, which will form the scrotum. So this urethral fold is what will now fold completely. Okay, and you are going to have something like that. So within it, you find a lumen of the penile urethra when urethral folds completely fuse. So this is what it's going to look like when everything is fine. That's the penile urethra. That's the glandular part of the urethra. That's the glans penis. Line of fusion of urethral fold is there. Line of fusion of scrotal swelling or labioscrotal swelling. But at a point, you can't tell whether the external genitalia is actually going to form a penis or clitoris. And the same way, the gonad too may not be seen as a testis or an ovary. Okay, so let's look at it. That is one, developing penis or clitoris. Urethral groove is number 2. The urethral folds number 3. So this is the fold that will join eventually, okay, to form the spongy portion, and then therefore you have the urethra within it, because this tube will now be found within, whilst the folds, merge. So that's what you had here. Once this fold gets completed, that space will now form the lumen of the penile urethra. Then that's the number 4, labioscrotal swelling, which will form the scrotum in male. Then number 5 is the anus. So if it develops along the male line, then this time around, number 1 will become the glans of the penis. Remnant of the urethra groove is number 2. It means that it has not completely closed. The urethral fold has not completely folded. And we will come to realize that if it is not completely folded until the fetus, becomes a neonate, then you are going to have issues with urine dripping from there. That's called the hypospadias. You have urethral folds in the process of closing, just like I said. So number 3, so the groove has not completely folded. Then you have number 4, labioscrotal swelling fusing to form the raphe of the scrotum, so the midline there. So these are going to meet and form the scrotum. The 5 is on. So this hypospadias, when the urethral fold do not completely fuse, then you have the urethral space being patent. Okay, it means that it is not covered ventrally, so it is exposed, for want of a better word. This is what you find abnormal urethral orifices because the urethral folds are not really fusing very well. So I'm sure we have established the whole background and foundation so we can answer these questions: genetic sex, chromosomal sex, gonadal sex, the effect of testis determining factor, what is SRY gene, what's an indifferent gonad, what structure marks or epitomize the indifferent gonad, what is tunica abuginia and state its basic function, especially in the male, then genital ducts, mesonephric, paramesonephric ducts, and their significance. Thank you very much.